So today we're going to go through some of the exciting things that we've been doing in the last 12 months or so. I'm an immunologist. I'm very much interested in understanding how the human immune system works. And my colleague Ellis is here to take you through how the data analysis side of things would work. And we are really fortunate to have Ellis working with us. So I'll give you a bit of a context to which we kind of do all these experiments. So as I said, as a human immunologist, I'm very much interested in understanding how the human immune system deals with infections and cancers. For a long time, we've learned a lot about the human immune system through understanding what happens in the peripheral blood, which is what I would class as a systemic response. But we realized that we needed to know what happens locally within tissues and tumors. So for the last 5 years or so, we've spent a substantial amount of time trying to develop protocols and to understand how the immune responses take place locally within tissues. And we were really fortunate. Sydney Cytometry core facility, one of the core facilities at the University of Sydney, decided to purchase Imaging Mass Cytometry. So we were one of the early adopters, and I had a very talented postdoc, Angela Ferguson, who spent a fair bit of time establishing the protocols, panels of antibodies to discover different immune populations and set up protocols for actually studying different human tissues. So that included, obviously, the wet lab work of establishing different panels, but also working with people like Ellis in establishing pipelines for analysis. I thought I would give you a simple example of a project, and that'll lead nicely into why we are interested in spatial transcriptomics. In this example, we are trying to understand the metastases into the lymph nodes of melanoma tumors, and we're trying to understand what the tumor immune microenvironment could do or how it could impact the clinical outcomes. So Angela has done a 40-panel IMC, generated the data. We usually do clustering, unsupervised clustering approach to identify different immune populations. And one of Ellis's PhD students came up with this really nice chat plot to look at an event and what it takes, the time it takes to the event, in this case, relapse, what possible cells or populations could be contributing to that event, whether it be immune or non-immune populations. So when you go through these data very carefully, you realize that there are obviously certain cell populations of interest when it comes to local responses. And you can take it a little bit further if you really want to understand how they are interacting with immune and non-immune populations. So that's something that we were really interested in very early on. And again, thanks to Ellis's team, we could put together a map of immune populations and non-immune populations that interact within the tumor microenvironment. And for example, you can actually see fibroblasts interacting heavily with a population potentially of macrophage type. But then we soon realized that this is wonderful. We can actually map the immune landscape. But what we really wanted to know was what those interactions truly meant locally and how they were impacting the clinical outcomes. Now, this interaction is based on proximity. But what really wanted to do is to go a little bit deeper in understanding what molecular pathways were activated on the cells that were interacting to gauge pretty much what those cells could be doing in that microenvironment. At that stage, we didn't have platforms that enabled us to run spatial transcriptomics and proteomics on the same tissue section. But we really wanted to see whether that's something we could do by combining spatial transcriptomics on Xenium platform with imaging mass cytometry. And that's literally what we've done. And I was really fortunate to have Ruth in the lab. She was completing a PhD and she was brave enough to embark on this journey to see whether we could actually run Xenium first and then run imaging mass cytometry followed by the Xenium on the same tissue section. And that's exactly what Ruth, with the help of Emily, has done here. And we've set up a protocol. We're in the process of putting together a manuscript that'll come online very soon, and the details will be there on that preprint. There were two concerns when we started doing this. First of all, we were not sure whether the tissue would hold through this process. We are putting it through two different platforms. But we really were fortunate to see that after Xenium, we didn't actually have massive tissue loss. We did lose some tissue, but we could retain a large number of cores quite intact. As you can see here, Ruth has done an H&E stain to show that the sections or the cores that were on the slides have held really well. So that ticked the first concern. The second concern we had was running Xenium prior to imaging mass cytometry, whether it would compromise the protein stains that we really wanted to do. So again, we were a little fortunate because we had used the same block here to run an IMC a year prior to running Xenium. So we actually had data from the original IMC run. So we also now have data of IMC run post-Xenium, and then we actually took a serial section of the TMA to do another run of the IMC so that we could actually compare the effect of running Xenium on the protein stains. As you can see quite clearly that running Xenium prior to IMC didn't seem to have compromised any of our protein stain, and we're giving you a couple of examples here, which was really promising. So that gave us the confidence that we could indeed run Xenium first, followed by IMC on the same tissue section. So then, obviously, the million-dollar question was, how does the data look when we compare the two different platforms? So to keep it brief, we looked at various different markers that are associated with immune populations. So at the top row, you can see the data from Xenium. The bottom row, you can actually see the data from IMC. And as you hopefully can appreciate, largely speaking, at the core level, there's a huge concordance between both platforms. So where you would find B cells, for example, from Xenium, you would also find the same populations staining for CD20 with IMC. Same holds true for T cell population. Not just phenotypic markers, you can also delve into proliferative markers like Ki67 or some functional markers. So that globally gave us a bit more confidence that you can actually find immune populations from both these platforms matching really nicely. Then we focus, not everything looks as perfect as that image shows you. We went deep, zoomed in at a single cell level. And that's when we started to see a little bit of discrepancy between the two platforms. So if you look at CD3, you would often find IMC picking up cells that were CD3 protein positive, but we did not see any transcripts that were picked up by Xenium. And you can actually see very nicely in this example here, you can actually find cells that were positive for both platforms, identified by both platforms, but in some instances, you only could see it in one platform, not the other. So that's where the complications started. So if, and we know this really well, biologically, we know not all proteins correlate with their respective RNA. We also know that there could be some technology-based challenges here. So that put us in a bit of a difficult situation in integrating the two data sets. So as I said, our ambition was to actually integrate the proteomics with that of transcriptomics, so we could actually really understand what is the molecular pathways that are activated when cells are interacting within the tissue. One of the things that we learned very quickly is rather than using one or single transcript and a single protein for identifying cell types, it would make sense to combine multiple proteins and multiple cell transcripts for an identity of a cell. That's literally what we have done, and I'm going to pass it to Ellis at this stage, who will take you through how the data analysis side of things happened. Over to you, Ellis.
Okay, so I actually really want to talk about this discrepancy between the transcriptomics and protein data, because I think that's one of the most exciting things about combining these two technologies. But before I do that, I just wanted to run over a few really simple statistical concepts and that makes it easy to appreciate these differences. And so to start with, when we had our tissue, just because we've got one piece of tissue we're going to be talking about, the question is, what can we learn from it? And so we can learn a few things. And so here we have phenotyped all of our cells, we've clustered them. And then on top of that, we've used a domain identification approach to segment or digitally dissect different regions of our tissue slice. And you can see here that at least region 4 is highly enriched for tumor cell populations. And so given what we know about these tumor samples, if we wanted to verify that our technologies were working appropriately, we could go and look for biology that we know exists. And so one of those things would be that we have seen previously that cytotoxic T cells, so the T cells that express CD8, there's more cytotoxic T cells in tumor than outside of tumor. And so after digitally dissecting our image into the cells that are inside the tumor and the cells that are outside the tumor, we can see that there is more CD8 expressed on the T cells inside the tumor. And so this is at the protein level. And so if you reflect back onto the slides that Mainthan was showing, the protein seemed very sensitive, protein everywhere, but the transcriptomics was quite sparse. And so we weren't necessarily surprised to see that when we went and looked at CD8A transcripts, there were much fewer of them. But then there was this really subtle, arguably, change where maybe there are a few more CD8A positive cells at the transcription level. And so this is starting to hint at some of the discrepancies we're seeing. Again, transcriptomics, we can deeply phenotype cells. And so one of the things that someone might be interested in asking is, as T cells get close to tumor cells, does CD8 increase? And so again, we can go and look at that. And at the protein level, we see this relatively strong effect where the closer a T cell is to a tumor cell, the more CD8 protein it has. But again, just because of the sparsity of CD8 in our data set, we don't see that relationship there. But I wanted to show you all of this because I think these concepts are important, because it all brings us to a point where we see a reasonable amount of a discrepancy between our Xenium and our IMC when it comes to cell phenotyping. And so we put a lot of effort into registering our IMC and Xenium, and I think done a pretty good job. But when we went to look at the concordance of the protein data and the transcript data, what we found is that there's a substantial amount of discordance. And so on this plot here, we've got T cells identified by 3 different T cell populations. One set of T cell populations that was identified by both Xenium and IMC. One subset that was identified only by Xenium, and one subset that was identified only by IMC. And so we saw this. We were worried that these were registration problems. This caused us to iterate consistently over improving that registration. But again, we're pretty confident that it's working well. And so one question is, well, what's going on here? Is this technical or is this biologically meaningful? So again, we can go and reflect on the biology that we know, which is there hopefully is more CD8 T cells in the tumor region. And so we went and digitally dissected our tumor region, and this is what we see. And so we can see that in our tumor region, there are more CD8 T cells than CD4 T cells. So that's fantastic. But given that we'd seen such a big discrepancy between cells that were labeled by IMC versus cells that are labeled by Xenium, we weren't necessarily expecting to see the same thing when we went and looked at Xenium label cells. And so fortunately, we did see this relationship. And so clearly neither technology is necessarily wrong in its labeling, but maybe one is more right than the other. We then went and subsetted this even further. And so what we ended up finding was that whether a CD8 T cell was labeled by IMC or Xenium, you'd always see more of them than you'd expect by chance in the tumor regions. And then what I think is quite interesting, and it speaks to something about the sensitivity of Xenium, is that if you had a cell type that was labeled as a T cell, but the CD8 positivity might be ambiguous, that had nothing to do with the spatial localization. So, of course, while I was talking and waving my hands around CD8, one of the great things about these technologies, you can measure many, many, many different markers. And so here we have an illustration that not only shows that we can identify various activation markers in T cells that are enriched inside the tumor versus outside the tumor. What I think is quite interesting is that we have a situation where if you have a cell population, a T cell that's only identified by IMC, you will also see an enrichment of these proteins inside the tumor, and not so much at the transcript level. And then if you have T cell populations that are identified only by Xenium, you'll see this enrichment at the transcript level, but not the protein level. And so this is leading us to think that there might be some really, really cool, potentially timing issues where we're seeing changes at the transcript level before the protein level, or vice versa, which I think really starts to justify measuring both of these technologies simultaneously. And so I'd just like to acknowledge everyone in Mainthan's team who's really pushed this along, particularly Ruth, who will hopefully come on soon and give us a big smile, and then Emily and Farhan and everyone else involved.